EPFL iGEM team.
You can't touch this. You can't touch this. You can't touch this.
Can't touch this.
Can't touch this.
Hi everybody, welcome. I'd like to start out this presentation by inviting you to take a minute and imagine. Imagine a world where synthetic biology is part of every one of your daily lives. Envision a future where a little sugar is sufficient to charge your mobile phones. I'd like you to believe in a tomorrow where technology is alive. At the end of this presentation, I'd like to show you that we, the EPFL iGEM team, were pioneers in bringing synthetic biology to technology. We'd like to show you how we were able to bring life to technology and technology to life for our groundbreaking project, the BioPad. The BioPad essentially is a fairly simple device. It's a living trackpad allowing the control of any associated electronic device. Basically, it's made out of three major components: a custom-designed microfluidic chip, engineered stress-responsive organisms, and a cheap signal detector. The process by which touch is detected is also fairly intuitive. When the microfluidic chip is touched, the stress-responsive organisms respond by emitting a fluorescent signal. Then, the signal detector catches the signal and processes it to allow the control of the associated electronic device. So at this point, I'm betting that most of you might be skeptical about one major point. How can biology be so fast? How can biosensors react to a signal so quickly? Well, if you'd asked me that question 4 months ago, I'd have told you were right. Biosensors are generally slow, and they're transcription-based. So stimuli detection in a matter of hours is totally normal. So we had a real problem in front of us. We had to build fast biosensors. So we came up with a novel solution that we'd like to call next-generation biosensors. Biosensors relying on post-transcriptional modifications to emit signals. Biosensors allowing stimuli detection in a matter of minutes. So the way we did this was fairly straightforward. We combined protein complementation techniques to dimerizing elements of a biosensor pathway. Essentially, what's happening is, upon the presence of stimuli, the dimerization of the elements leads to the reconstitution of the fluorescent proteins, thus signal emission. So having taken a look at how we were going to engineer biologically our system, let's take a look at our physical interface. We chose microfluidics as our physical interface for two main reasons. First, just as pixels on a screen, microfluidics allowed us to subcompartmentalize the signal emission by our organisms. Moreover, to induce mechanical stress on our cells, microfluidics was perfect because its main component, PDMS, is fairly flexible. So having taken a look at both the biological aspects and physical aspects of our project, we started our summer out with 4 main objectives. First, we wanted to develop a next-generation stress sensor, a stress sensor reacting to a variety of types of stress in a matter of minutes. We first wanted to characterize it through chemical stress, and then implement it through mechanical stress. Then, we wanted to design an optical microfluidic chip, a customized microfluidic chip that would be perfect for the BioPad. Then, we wanted to build a real-time signal detector, and finally, we wanted to put all these components together to build the first BioPad.
So throughout the project, we engineered stress biosensors in 2 organisms, E. coli and S. cerevisiae. As a first step, we engineered these organisms and characterized them through chemical stress rather than mechanical stress, as it's much easier to quantify. Of course, for our project, we need to induce a signal through mechanical stress, which is going to be our second step. I'm going to show you now how we engineered our E. coli strains and characterized them through KCl stress. So the stress-responsive pathway in E. coli that we used is called the CPX pathway and is composed of a sensor kinase, the CPXA, which phosphorylates its relay protein, the CPXR. When phosphorylated, CPXR homodimerize and become active transcription factor. We hijack the system by fusing split reporter to the CPXR. As you can see, stress induces the CPXR homodimerization, leading to the reconstitution of the reporter protein, thus a fast signal emission. When choosing a reporter protein for our system, we came up with a newly developed split infrared fluorescent protein, split IFP, a Nature publication from April this year. So the split IFP has the advantage to be reversible, a vital characteristic for our biopad, as we need to both turn on and off our system. The split IFP also has the advantage to trigger very low background noise, as it emits in the infrared spectrum. So we fused our IFP1 and IFP2, whether at the C or N terminus of the CPXR, as we didn't know which orientation would lead to the reconstitution of the reporter protein. So here you can see one orientation when both IFP1 and IFP2 were fused to the C terminus of CPXR. And here you can see all the other configuration that we designed this summer. We tested our 4 constructs on a plate reader under KCl stress. We measured the relative fluorescence normalized by DOD in function of the time. And you can see that the only configuration leading to an IFP signal is the one when both IFP1 and IFP2 are fused to the C terminus of the CPXR, and that the other configuration don't lead to any IFP signal, nor the negative control. If we zoom in at the point when we added the stress, you can see that the IFP signal increases immediately after the induction of stress. We were also able to visualize our IFP signal under a microscope, 100 times magnification, and an APC filter. On the left, you can see unstressed cells, and on the right, 50 millimolar KCl stressed cell after 5 minutes of stress induction. You can see that the difference in signal is pretty important and very fast. For our biopad, we don't only need to activate our signal, but also to deactivate it once the stress source is removed. So we launched another experiment on the plate reader under KCl stress again, and after about 2 hours, we removed this KCl stress. And you can see that the signal drops drastically. So at this point of the project, we were able to both turn on and off our IFP signal in E. coli in a very fast way. So we were pretty happy about it because in vivo studies has never been done before about CPXR homodimerization, and also because it was a big step ahead for our biopad.
All right, now for the yeast part. We first of all also wanted to characterize our system using chemical stress. Now, the pathway that we engineered in Cerevisiae is called the HOG pathway. And the principle is pretty similar to what we had before with the CPX pathway in bacteria. So upon osmotic or mechanical stress, we have the interaction of 2 proteins. PBS2 is going to phosphorylate HOG1. And we use this interaction to bring together our 2 split reporters. For yeast, we chose the superfolder GFP. Now, once our constructs were ready, we did this experiment where we stressed our yeast with acetic acid. So we see on the graph here that we were able to obtain a great increase in fluorescence upon the acetic acid stress compared to the yeast that were not stressed. Now, we were curious about these yeast cells and brought them under the microscope. And again, here we see the pictures where we have the non-stressed cells and the acetic acid stressed cells. So we have a clear difference in GFP intensity. So at this point, we achieved to have a chemical stress responsive organisms both for E. coli and S. cerevisiae. And the next step now, as we want a biosensor that is touch responsive, was to test the mechanical stress. For this, we used microfluidics. And due to practical and time issues, we chose to focus on our E. coli construct with the IFB reporter. So this is our microfluidic biopad interface, which essentially is composed of 6,400 chambers interconnected by these channels. These channels allows us to bring the cells inside the chambers and also to bring the needed media to culture the cells on chip. Now, I would like now to discuss a little bit the evolution of our designs, which led to our final interface. Because throughout the summer we went through different chip designs, and we started off by using an existing design called Maitomi. But this chip, we wanted to be sure that we are able to culture cell on chip. Next, we also wanted to induce reporter genes. So, as our biopad should contain living organisms, our first aim was to be able to culture cells on chip. So here you see a single chamber where we did an overnight culture of E. coli. So next step, we wanted to be able to induce a reporter. So in this case, we used bacteria transformed with SFGFP that we can induce by arabinose. We float the cell in the microfluidic chip and added arabinose on the upper half of it. As you can see here, we had great signal on the upper half. The lower half does not have arabinose, thus no GFPC. We quantified this experiment and obtained this graph. Now, next step was to create our own smash coli chip. This chip, we wanted to use it to, first of all, see if we are able to measure an IFP signal on chip. And second of all, and most importantly, we wanted to be sure that we can test the mechanical stress on chip. So that's the smash coli design. It's a chip composed of 224 chambers on which we can apply mechanical stress by using so-called buttons. If we zoom in here, we see these two chambers, and if we pressurize this little square here, this is the button. If we pressurize the button, we are able to squish the cells underneath. Now, we used this chip extensively this summer to optimize our constructs. So as I said, first of all, we wanted to be sure that we can measure IFP on chip. So what we did here, we took our bacteria, stressed them with KCl, and brought them inside the chip, and we measured. So here we have the non-stressed cell, and here the stressed cell. So with this experiment, we were able to show that we are able to see a difference between stressed and non-stressed cells for an IFP intensity. Now, next step was, of course, the mechanical stress experiment. And what we did here, we followed our cells in the microfluidic chip and applied the buttons, as I just showed you. Now, what we obtained here, these two promising results, where we see that before pressure induction and after pressure induction, we obtained this difference in intensity. Now, this picture was taken within 10 minutes after the pressure induction. Okay, at this point, we're almost ready to create our final interface for our biopad, which would look like that. And basically, this interface should simply be able to transfer the pressure that is coming from your finger on the cells underneath. So at this point, we can take our two first objectives, which were to develop a next generation stress sensor and design an optimal microfluidic interface.
Now, what was left to do was to engineer a real-time signal detector. We did that in a way that we would simply put a lenses underneath the biopad that would focus the light onto a camera, which then transmits the signal to a Raspberry Pi, a small signal board computer. Now the hardware, all of this costs around $150, which is really not much for lab gear. Then on the computer, there would be our customized program that would process the signal in the following way. In this video, you see how the flashlight is tracked. Mainly, it's the maximum intensity of a certain wavelength that is followed. Now, knowing that the program works, we needed to know, is it possible to track also much lower light quantities as biofluorescence? So we loaded some cells that would express superfolder GFP on our chip that then would be excited with a blue LED light. And here on the right side of the video, you see that we were able to track the signal of the bacteria with our program, which is quite exciting. Now, so we were also able to do that, and now we're in a really good position to actually build the biopad. Now, as it comes to lab safety, we have the same problem as a lot of other iGEM teams if we want to take our device outside. All of us, also if it would be some kind of detector for pollutant or something, we put our modified organisms onto the chip, flow some media in there, but we cannot guarantee that nothing comes out. And outside of the lab, this is a no-go. So after brainstorming and putting our heads together, we came up with a solution for that. We called it the Clean Coli. Now this is not a chip, it's more like an extension for any existing chip with 4 decontamination steps. First of all, we would lyse the cells, and then to really be sure that they're all dead, we would bleach them. Then they would go through a filter, just filtering out anything of the liquid. And because we really want to be sure, we have a microheater that heats it up to 95 degrees Celsius. So anything that comes out is clean. Now, having done all of this and having thought of this, we wanted to know, what do other people think of synthetic biology, iGEM, and especially our project? So we would send a quiz to 100 high school students, but as they came back, what we learned were they were just thinking of vegetables and how they would get bigger, but not necessarily better. So what we did, we invited them over to our EPFL campus where we had a special program for them. First, they would have a general presentation about iGEM, our project, and synthetic biology. Then they would go into a lab workshop that we prepare for them, and in the end, they would develop their own iGEM project, which is pretty remarkable because they did not have the same background that we do, but they still came up with projects that other iGEM teams were actually doing. Now, all of this would not have been possible without our sponsors, EPFL, and our advisors. And at this place, I would especially like to thank Roshana and Oleg, who are not here today because of visa issues. And to you, I'd just like to say that I hope we were able to show you that the biopad is something that tomorrow you can touch. Thank you.